A single bacterium cannot be seen with the naked eye, yet within days it can form a visible colony containing millions of descendants. This transformation relies on precise molecular mechanisms rather than magic. Understanding how individual cells coordinate division and resource acquisition reveals the physical constraints governing biological expansion. After that first surprise, we can slow down and trace the mechanism behind from one bacterium to millions, how colonies grow from the first physical step, so the effect stops feeling mysterious and starts feeling engineered. We begin by establishing a fundamental distinction between an individual bacterium and the colony it forms. A single rod-shaped cell is microscopic, requiring high magnification to observe its structural details. In contrast, a bacterial colony appears as a visible white circular mass on an agar plate surface. This macroscopic structure arises exclusively through clonal expansion of that initial progenitor cell. Every cell within the colony shares identical genetic material derived from repeated binary fission events. Consider the difference between viewing one isolated bacterium under high magnification versus observing the textured surface of a mature colony. The colony represents a population unit, where biomass accumulates until nutrient depletion or space constraints halt growth. Understanding this structural definition prepares us to examine how these populations acquire essential resources for continued expansion. Having defined the colony as a product of clonal expansion, we now examine how these populations acquire essential resources for continued growth and division. Pause 0.8 to transition from a single bacterium to a macroscopic colony. The cell must first overcome physical constraints imposed by its environment. The primary challenge involves acquiring sufficient carbon sources through passive diffusion and active transport across the lipid bilayer. Passive diffusion relies on concentration gradients, where molecules move spontaneously from regions of high density toward lower density without energy expenditure. For example, small nonpolar molecules like oxygen traverse the membrane directly by dissolving into the hydrophobic core of the phospholipid layer. In contrast, polar nutrients, such as glucose, require specific transmembrane proteins to facilitate their passage through the otherwise impermeable barrier. These transporter proteins undergo conformational changes that allow substrates to bind on one side and release them into the cytoplasm on the other. Active transport mechanisms further enable uptake against a gradient by coupling molecular movement with energy derived from ATP hydrolysis or ion gradients. The rate of nutrient flux is strictly determined by the steepness of the concentration difference and the density of functional transporter proteins embedded in the membrane. Without these precise acquisition strategies, intracellular metabolic pathways would stall due to substrate depletion regardless of external abundance. Once sufficient biomass accumulates through this regulated uptake, the cell reaches a threshold that permits entry into the binary fission cycle. Pause 0.8's binary fission begins when the bacterium initiates DNA replication at a specific origin point on its circular chromosome. Proteins assemble sequentially to unwind the double helix and synthesize new complementary strands, ensuring each daughter cell receives an identical genetic copy. Once duplication is complete, the nucleoid regions separate as the cell elongates, creating space for two distinct genomes. Simultaneously, a protein ring forms at the midline of the cell, recruiting enzymes that synthesize new peptidoglycan to build the septum. This constriction process physically divides the cytoplasm and membrane, resulting in two independent daughter cells from one parent organism. For example, under optimal conditions, Escherichia coli can complete this entire cycle approximately every 20 minutes through precise molecular coordination. The rate at which this division cycle occurs depends heavily on environmental factors that regulate metabolic activity. Pause 0.8's temperature and pH act as fundamental variables that directly modulate molecular motion and structural stability within the bacterial cell. As environmental temperature rises, kinetic energy increases, causing molecules to vibrate with greater intensity and frequency. This heightened vibration can distort the three-dimensional shape of enzymes, specifically altering the conformation of their active sites where catalysis occurs. When an enzyme loses its precise geometric fit for a substrate molecule, metabolic reactions slow or cease entirely, reducing overall growth rates. Conversely, extreme acidity or alkalinity disrupts hydrogen bonding networks that maintain protein integrity and membrane fluidity. Consider the contrast between mesophilic bacteria thriving at moderate temperatures versus thermophiles adapted to high heat. Their enzymes possess distinct structural stabilizations to withstand thermal stress. Visualizing this relationship reveals a bell-shaped curve where metabolic activity peaks within an optimal range before declining sharply under thermal extremes. These physical constraints dictate the boundaries of viable growth zones, setting the stage for how populations organize themselves in space. Pause 0.8. We begin by examining how individual bacterial cells organize themselves into a structured three-dimensional community rather than remaining as isolated entities. The extracellular polymeric substances function as a physical scaffolding material that binds cells together and maintains the colony's structural integrity against environmental forces. This matrix creates distinct layers, where packing density varies significantly from the surface to the interior of the colony structure. Cells located in densely packed regions experience restricted diffusion rates for essential nutrients, while simultaneously accumulating metabolic waste products at higher concentrations compared to peripheral cells. Consider a three-dimensional cross-section rendering that reveals how oxygen gradients form as consumption outpaces supply within the core layers of the colony. This spatial heterogeneity establishes microenvironments, where different physiological states coexist depending on local resource availability and physical constraints imposed by neighboring cells. This spatial organization sets the stage for understanding how population density signaling emerges as cells communicate across these structured microenvironments. Pause 0.8. Quorum. Sensing is a concentration-dependent chemical accumulation process, where bacteria communicate through secreted signaling molecules. As individual cells divide and multiply within the colony, they continuously release small organic compounds into their immediate environment. These molecules diffuse outward but accumulate locally when population density increases beyond a critical threshold. Once sufficient concentrations are reached, the ligands bind to specific receptor proteins embedded in bacterial cell membranes. This binding event triggers intracellular signaling cascades that ultimately alter gene expression patterns across the colony. For example, in vibrio fishery populations, autoinducer molecules accumulate until they activate luciferase genes for bioluminescence production. The contrast between low density and high density states demonstrates how identical genetic material produces different phenotypic outcomes based on environmental concentration. This mechanism allows coordinated behaviors that would be ineffective if performed by isolated individual cells acting alone. Pause 0.8. As the bacterial colony expands through binary fission, every single division event consumes nutrients and releases metabolic waste into the immediate microenvironment. Unlike multicellular organisms that possess circulatory systems to remove toxins, individual bacteria rely entirely on passive diffusion to clear these byproducts from their surroundings. Consider a dense cluster of Escherichia coli cells where sustained respiration generates organic acids as a primary waste product. Over time, the concentration of these acidic molecules rises within the colony center faster than they can diffuse outward into the surrounding agar medium. This localized drop in pH alters the electrochemical balance required for essential enzymes to maintain their functional three-dimensional structures. When critical proteins denature due to this chemical shift, cellular machinery halts and division ceases regardless of available nutrient reserves. The heat map visualization demonstrates this phenomenon by showing a steep concentration gradient, where red zones indicate toxic thresholds that inhibit further replication. Pause 0.8. We now synthesize these individual cellular behaviors into a population-level growth curve that maps directly to specific biological constraints. The initial lag phase represents cells adapting their internal machinery, where metabolic pathways are reconfigured without net division occurring yet. This transitions sharply into the log phase once sufficient ribosomes and enzymes accumulate, allowing exponential replication driven by abundant nutrients. As resources deplete, waste products accumulate to toxic levels, forcing the population into a stationary state where birth rates equal death rates. Finally, when inhibitory conditions overwhelm cellular repair mechanisms, the death phase emerges as net loss of viable organisms becomes inevitable. The sigmoidal pattern demonstrates that physical limits on expansion are not arbitrary, but arise from specific mechanistic bottlenecks in resource availability and chemical toxicity. This establishes the physical boundaries of expansion, which we will now examine through the lens of environmental carrying capacity. Pause 0.8. This script establishes that bacterial growth is a measurable physical process driven by specific cellular mechanisms. Each section builds from molecular interactions to population-level patterns without invoking unexplained phenomena.